There are different kinds of cell reproduction. We're going to look at 3 specifically. The first kind we're going to explore is called binary fission. Bi means 2. Fission means to split. And this is a kind of cell division where prokaryotic cells, if you remember, prokaryotic means cells that do not have a nucleus. Pro means before, karyotic means nucleus. These are cells that evolved before the nucleus evolved. We're going to look at how they do this simple kind of binary fission where they grow to twice their size and they split into 2 new cells. Then in eukaryotic cells, and I don't have this specifically on here, but maybe you want to put a little note here, that mitosis and meiosis are kinds of eukaryotic cell division. And we see we have 2 different kinds depending on what kind of cell we're looking at. Mitosis is a kind of cell division in what we call somatic cells or non-sex cells. When we're talking about non-sex cells, we're talking about everything in a body that is not an egg or a sperm. Meiosis, it only talks about the sex cells and the making of gametes. The term gametes refers to egg and sperm, or we might call them sex cells. So we're going to take a look at those 3 different kinds of cell division. Let's talk a little bit about sexual reproduction versus asexual reproduction.
Where would I find this on here?
Right here.
Oh, okay.
Right at the bottom. Yeah, right at the PDF of the notes are right at the bottom of our homepage and Canvas. It's just like a little file right at the bottom. Okay, so let's talk about sexual versus asexual reproduction. Sexual reproduction, when we're talking about sexual reproduction, it's today especially, it's not going to be as sexy as you might think. When we're talking about sexual reproduction, we're talking about the creation of gametes. Gametes are, again, egg and sperm, or the sex cells. Gametes are made through meiosis, not mitosis, meiosis, and these cells are genetically unique to one another. And we'll talk about how we get there as well. Asexual reproduction comes from a single parent cell. You don't need to have a biological male and female. It's just one kind of cell. So single parent, no need for another individual to do asexual reproduction. No sperm, no egg. When we're talking about eukaryotic cells, we're talking about them being produced through mitosis. And offspring are genetically identical to the parent for the most part. Sometimes we get mutations, but those are very minor. All right, so let's take a look at this term zygote. Zygote is the first cell. When an egg and a sperm merge together, that very first cell that the egg and sperm combining make are called a zygote. Sometimes we also call it an embryo. Zygotes are formed through sexual reproduction. So here we have what's called an oocyte. An oocyte is an unfertilized egg. We have a fertilized egg. You can see the sperm merging with our oocyte. And when their DNA combines together, when the sperm and the egg's DNA, the genes or the chromosomes come together, you form what is called the zygote. That's that very first cell. And then we will eventually get into, well, oh no, we don't get into this in this class. So, and then they go on to become larger cells. The term diploid, di, means 2, just like bi means 2. So if you might want to make a note that di means 2 there, also bi for binary 2, di for diploid means 2. That diploid means that a cell has 2 sets of chromosomes. The zygote is diploid. So when we take a look at our little illustration here, that when you have what's called a, we'll talk about haploid, and haploid means 1, 1. When you take a haploid cell, the sperm, and a haploid cell, the egg, and you add 1 plus 1 together, one genome. Term genome, which we're going to get to defining, but just to let you know, a genome are all of the genes that an individual contains or a cell contains. So when we take the genome, all the DNA, of the sperm and all the DNA of the egg, 1 plus 1 equals 2, or haploid plus haploid equals diploid. So when we take a look at this is haploid, the egg or the oocyte, the sperm, haploid, and when they come together, what they make are the diploid zygote, 2 sets. And so when we're talking about 2 sets, you can talk about 2 sets of DNA, a set from the biological mother in the egg, a set from the biological father in the sperm, 2 sets of DNA, 2 sets of chromosomes, or 2 genomes come together. So that when we're talking about diploid 2, di means 2, 1 haploid plus haploid equals diploid. When we take a look at what's called a karyotype, this here is a human karyotype. A karyotype is where they can manipulate a cell during, we'll talk about cell division, mitosis specifically, to lay out all of the chromosomes. And so it's like super cool. Somebody can go in, split open the nucleus of an egg that's of a cell that's undergoing mitosis, and they can take all the little chromosomes and they can place them and find the 2 that are alike that we call chromosomes number 1, the 2 number 2s, the 2 number 3s, 2 number 4s, and they can place them into pairs. And the reason why you have 2 of each kind, does anybody know why you have 2 of each chromosome? Where do they come from? Yes, good. 1 from mother, 1 from father. Yes, very good. So you have 2 of each kind of chromosome because you got a chromosome number 1 from your biological mom and a chromosome number 1 from your biological dad. So why you have 2. What we call those 2 chromosomes of each kind are homologous. H-O-M-O in biology, in general, it means same or alike. Just like if you are homosexual, you date someone who is alike you, same or alike. So we use that word or that beginning of the word in our society as well. Homologous chromosomes, 2 of the same kind. So you get 2 because you get 1 from biological mom, 1 from biological dad. The thing about these homologous chromosomes and how they know to place them, now in our cells, they're not like rainbow colored like this. These are actually dyed so that it shows you very clearly that the number 1s are the same, the number 2s are the same. I mean, the 2s don't look exactly the same in this picture, but they are, when we're talking about homologous chromosomes, they are the same length. They have the same amount of DNA in them. And so that's why they go together. They have the same gene banding patterns. So for example, let's just say, I'm just making this up off the top of my head, that on chromosome number 1, again, making this up, that on each of the 2 chromosomes, you have a gene, what we call location or loci, and that loci, it codes for handedness, whether you are right-handed or left-handed. From each of your parents, you get a gene for your handedness. And so there are possibilities of those genes. You could get a right-handed gene and a left-handed gene. You could get 2 right-handed genes, or you could get 2 left-handed genes. So while you have the same band or location, loci of that trait, where that gene for handedness is located, you can have different versions of those genes, and those are called alleles. And again, we're going to define that in our notes eventually. Okay, oh, right here we are. Okay, so the gene location or loci, it is the location of a gene on one of those homologous chromosomes. Oh, so as I just defined that. And then back again to our karyotype. That our karyotype is when we lay out all of our chromosomes in the full chromosome form. And we can see from an individual, one of their cells, what all of their chromosomes look like. Our 23rd pair, our 23rd homologous pair, will tell us whether we are XX for a biological female or XY for a biological male. So this is the one case in which we have a homologous pair, and half the individuals in our world will be different. They won't be the same. One will be an X and one will be a Y. Otherwise, the biological female is two X's. And then we'll talk about things that go wrong in the world later in this unit. Okay, so again, haploid. Haploid is half the amount of chromosomes in a cell. Haploid means one. Diploid means two. Egg and sperm has one set of chromosomes each. 1 plus 1, haploid plus haploid equals 2, diploid. And again, you can see haploid sperm, haploid egg, 1 plus 1 make our diploid zygote. All right, so defining genome. Genome are a complete set of all of the genes of an organism. If you find any mistakes in the packet, please let me know. I'm always going back and forth between the PowerPoint and the packet to make sure I have everything. Sometimes I miss pasting something in. So please, if you see any discrepancies, let me know. A gene is the physical unit of heredity. It tells you, your genes tell you what you're going to look like, how you're going to act. Our behavior is passed on in our genes, and how we internally function or our metabolism. Yes, it does. And I think somewhere in your notes, I know I address it somewhere, somehow in your notes at some point. Yes. So mitochondrial DNA is part of our genome. So if you didn't know, in addition to our nucleus, thank you for bringing that up. That's a great addition. Our mitochondria and our cells, little powerhouses that convert our energy of our sugars and our other foods like carbohydrates and lipids. They go into metabolic pathways to go into cellular respiration, which is performed by the mitochondria. The mitochondria has DNA that comes from our maternal side, from our mother. We receive that from the oocyte or the egg of our mother. We get her mitochondria. We do not get any mitochondria from sperm from the father because the sperm is only carrying, it only has like 4 things or 4 parts, and it wants to swim, and we'll get to that too. It swims a really, 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 really long way, so it doesn't carry any organelles that it doesn't need. So the egg, it carries our mitochondria. And again, our mitochondria has some DNA to it, and so they can go back over many generations and look at the maternal inheritance over time to see what has been passed on from your maternal or the female lineage of your family. An allele, as I talked about handedness, for handedness, we have 2 alleles, 2 possibilities for that gene. We have right-handed allele and left-handed allele. They're both kinds of genes that code for handedness. And so when you have different versions of a gene or a trait, we call those alleles, alternate versions of a gene. The trait is the description of the gene. When we're talking about handedness, the trait, we say handedness is the trait I am talking about. The alleles are the different versions, right and left, for that trait. And when we're talking about a trait, when we take the genes, and we'll get into this like in a little, we'll get into it, no, we'll get into it on Wednesday. That we have different genes or alleles for handedness. We can get a capital R from biological mom, a capital R from biological dad that will code our trait to be right-handed. If we get a capital R from biological mom and a little r from biological dad, this still codes for us to be right-handed. The only way we're left-handed is if we get a gene for little r for left-handed and a gene for little r for left-handed. You have to get one from each parent in order to be left-handed. When we take these and we interpret them into the physical description, we call that a phenotype. Let me see if I wrote genotype down here. No, did I? No. So these are the genotype. And this so the actual genes are the genotypes. The description, the physical description of the genes, right-handed or left-handed, are phenotypes. Did you want to add something or question? Oh yeah, okay. Okay, so prokaryotic cells, they have, in terms of their genome, if you remember back to studying the structures of the prokaryotic cell, the cells that do not have a nucleus, they have their DNA here. It's called a nucleoid. Nucleoid. The nucleoid chromosome is the main DNA of a prokaryotic cell. If we took all this like squiggly stuff and we stretched it out, it would be a big circle. It's a circular chromosome. It goes in one circle. Our DNA, our chromosomes, they start at one end and they kind of twirl around like this, and there is an end to end. It's very different in eukaryotic cells than a circular nucleoid chromosome. Now, some prokaryotes can have additional DNA. Yes, good, plasmids, exactly, very good. So they can also have these little circles in their DNA, in their cytoplasm. So prokaryotic cells, like how we can have our extra DNA, or we do have our extra DNA in our mitochondria. Some, not all, prokaryotic cells, some have plasmid DNA that are little tiny rings that are in the cytoplasm. Yeah. We are going to do some genetic engineering. I have to find out what that lab is. I am not sure yet, but we will find out as we go. I have not taught Bio 111 since before the pandemic, so I used to teach it every semester. I know some of the labs have changed, and because it's summer and I've been busy and had a lot going on, I'm catching up with you all, but I will know how to do everything day of. Yes, but we're going to do some genetic engineering, yes, which is exciting. Interesting thing about these plasmids is that the plasmids are very good for the survival of prokaryotic cells. On those plasmids, they may carry genes for resistance to antibiotics. If you've heard about MRSA, MRSA, or any of the super bugs that are out there, they have very wide spectrum antibiotic resistance, which is great for them, right? It keeps them alive, but is that good for us? No, not good. If you are interested in medicine, pharmacy, genetic engineering, this is a huge field of study that needs brilliant problem solvers to go into it. because we need to figure out better antibiotics. Because these plasmids, what can happen is that one species of bacteria, so you can see this one here, it's kind of elongated in shape, and it has these little fuzzy things these little hairy things called pili. Bacteria that are totally different shaped, different species, they can share plasmids with each other. So here's a very fascinating thing that's happening with prokaryotic cells, is that they can share DNA with species that are different from themselves. They can do that naturally. We can't do that naturally. We can get genes from other organisms, but we have to do that like a whole genetic engineering thing to do it. But they can do this naturally. And that's why over evolutionary time that prokaryotic cells have gotten more resistant to different kinds of antibiotics, because this one could have like antibiotic resistance to penicillin, and this one could have antibiotic resistance to streptomycin. And even though they're different species, they can give each other that. So now this one has penicillin and streptomycin resistance, and this one has penicillin and streptomycin resistance. Crazy. That's nuts. And we're in a big bind because there's not a ton of money that goes into this, because prokaryotic cells are developing and evolving at a much quicker rate because they have very short evolution times, because their generation times are very quick. Prokaryotic cells can replicate or reproduce in about 20 minutes. New generation every 20 minutes. And each of those new generations can produce new generations. And so the mistakes that can be happening when they are copying their entire genomes super quickly in 20 minutes, do you suppose if I said, you need to copy this whole thing in 20 minutes, do you think you'd make mistakes? Would you be stressed out? Yeah, you'd be stressed out, right? And so if you're thinking about a prokaryotic cell, their volume of their genome is essentially like this much information that they are replicating or copying to a new generation in 20 minutes. The possibility of mutation or them making an accidental mistake and the copying of that genome is high. Some of the mutations or accidental mistakes that they make are more resistance to more different antibiotics just by chance. They don't try and do it. It just happens totally randomly by chance. And so we are in a lot of trouble because they can replicate quickly. They can share. Pharmaceutical companies, because antibiotics are becoming like outdated so quickly because of these issues, there's not a lot of money to be made. They have to keep doing more research and more research and more research, whereas like Viagra or insulin, which we know are solid ways to treat, like Viagra, right? It can treat the flaccid penis. We know that insulin can treat diabetes. They can just pump that out. They have one recipe and they just pump it out, pump it out, pump it out. And they make tons and tons of money, and they don't really have to do much. Somebody just kind of like monitors the process. Whereas this takes a lot of research. And so they have to put a lot of money into this. And they're like, we don't really want to put a lot of money into this. It's not the right thing. It's not very ethical, but they don't care. They want to make money. So there's a lot to be discovered in the world. Yes, exactly. So he said, we're not supposed to take other people's antibiotics. So let's say that you get some kind of bacterial infection, and one of the problems is that let's say you have a bacterial infection, you're taking penicillin for it, and it says, take this for 10 days. What people often do is that they'll take it for 5 days and they'll start feeling better, and then they'll say, well, I'm better. I don't need to take the rest of it because I'm better. But you may not have knocked out all of the bacterial infection completely because you didn't finish up your penicillin. And so then you have those in you. And then let's say it was for a strep throat and you cough, and then you could pass on the strep bacteria to other people in the world. And those bacteria could be highly evolved and also be resistant to penicillin. So there's like, they could have mutated, and there's, yes, so like it's very simple yet complicated. So if you ever have to take antibiotics, take the full amount of time for them, finish up that thing. But also, if somebody takes their penicillin for 5 days, they got a strep throat, they're like, I'm totally better. And then let's say your sibling also now gets strep throat that you probably gave them. And they're like, well, I'll take the other 5 days because you are better in 5 days, and then I can be better in 5 days. And so what that can do is it allows for evolution of the bacteria growing inside of you and your sibling now. So you never want to take someone else's antibiotics either. You want to finish yours and never take someone else's.
Isn't strep throat especially dangerous because if it's not taking the antibiotics right, there's a chance that it could cause mental illness?
There's so many, yes, absolutely. And things are, because of the evolution in these plasmids and quick generation times, there's a lot more crazy things that are evolving in prokaryotic cells, yes, which can also, like cause mental illness. In eukaryotic cells, the genome is found in the nucleus. The evolution of a nuclear envelope protects, so here's kind of an interesting thing, is that the DNA in our cells, because we are eukaryotic, meaning that we do have a true nucleus, we have a double membrane protection around our genome. It cannot get damaged or mutate in our cytoplasm by some kind of like cellular waste product. We have it protected within our nucleus. Whereas if we're looking at them, they could have like some of their little organelles could be spewing out metabolic waste products that can degrade their DNA and cause not a mutation naturally, but a mutation by kind of like a side effect. So there's another way that mutations are fostered in prokaryotic cells. And then again, you can see, so this is a prokaryotic cell with a cell wall, something like a plant or a fungus or maybe some kind of a protist. And this is one of ours. Could be different kinds of cells that are round, but this is essentially an animal cell. Also has a nuclear membrane. Okay, so we're going to talk about the very simple kind of cell reproduction in prokaryotic cells called binary fission. Again, remember, pro is before, karyotic is nucleus. These kinds of cells evolved before the nucleus evolved. They evolved about 3 to 3.5 billion years ago. So prokaryotic cells have been around a real long time, which gives them a lot of time for evolution in addition to their quick generation times and sharing of plasmids. Bi means two, fission means to split. So what we know about this kind of cell division is that these cells can split into two very simply. All right, so what happens is that what we call daughter cells, you have a parent cell and daughter cell. And the parent cell will grow in size to form two daughter cells. So just making sure you're on track. Fission means division. Fission, division, split in two. Get daughter cells from a parent cell. Essentially, and not in reference to maybe a mutation that might happen in the copying of the DNA. They are essentially genetically identical, but again, as I mentioned, when the parent is making that DNA, the genomes for the two daughter cells very, very quickly, they can, by accident, make mutations that are passed on. Before any kind of cell division happens, binary fission, mitosis, meiosis, the DNA has to replicate because you're going from one cell. They only have one genome. They have to replicate their genome to make 2 genomes that they give to each of their daughter cells. So any kind of cell division, DNA, before it happens, the DNA has to replicate. Do we have it in detail?
Like, for example, the helicase and the ligase and the Okazaki.
We're gonna go into that in the last unit. Yes. When we're talking about prokaryotic cells, remember again, their genome is that circular, even though it looked like a bunch of squiggles in the middle. If we stretch it out, that nucleoid would be a big circular piece of DNA. It is often attached at one point to the plasma membrane on one side of the cell. And that's pretty much it. They just have to, the other thing to remember is that often we don't put in these details of these kinds of fission. They also have to replicate all of the organelles. So not only does the DNA have to replicate, but all the organelles must replicate so that they have 2 essentially identical cells to themselves. We can see eventually that they will split into 2. These prokaryotic cells are round. Not all prokaryotic cells are that kind of long cylinder shape. Some are round, some are spiral shaped. They have all different kinds of weird shapes. We just usually focus on the very basics when we're talking about this class. If you get into, like, if you take a microbiology class, you'll learn a lot more about the diversity of shapes and sizes and things like that. Okay, so to sum this all up, we have a parent cell. Our parent cell is all their organelles. They have their nucleoid. It's attached to the plasma membrane on the inside. Before going into binary fission, see the nucleoid? Boop, boop, makes a copy. So it's going to pass on one genome to one daughter cell, one genome to the other daughter cell. It also replicates all the other organelles. It grows in size so that it accommodates the size of 2 new cells. You can see here, the nucleoids are replicated, the size grows. They are going to eventually pinch in the plasma membrane and then form a new cell wall between the 2 eventual individual daughter cells. Yes. All right, so now we're going to talk about eukaryotic cell division. We're going to look at mitosis first, and then we're going to go look at meiosis. So mitosis. Mitosis, the importance is for growth, repair, development, and maintenance. You all are in your adult phase of life. You're really doing more so repair and maintenance at this point. Evolutionarily, you are very old in comparison to our ancestors. Like if we went back even like 1,000,000 years ago, y'all would be like elders in our society, and I would be dead. So that's why adulting, as we get into, about in your late teens, definitely in your 20s. For some of you who are lucky enough, your metabolisms will stay high your whole life. But generally, our metabolism starts to slow down at some point, usually about your 20s, mid-20s, about 27-ish on average, because we just weren't meant to live this long. But we have a lot of interventions from our different kinds of technology, especially medicine. Like just think about, there's people who right now are experiencing heat waves that don't have air conditioning and they are dying of heat, right? So if you could get those people who are in those really, really hot areas, you could get them just cooled off, they would be fine. So we do have some interventions, but we don't have them available to every person on the planet, which is, that gets into political issues. All right, so when does mitosis kick in? Mitosis kicks in as soon as you have a zygote. Sperm and egg are made through meiosis. And when sperm and egg come together and they form that first cell, the zygote, then the zygote goes from one cell, mitosis to 2 cells, mitosis to 4 cells, mitosis, until you get to be us, So here has an illustration of this, that these, the only thing on this illustration, the cells that are made in meiosis is this. We make sperm and egg in our bodies through meiosis. Everything else, as soon as we get that zygote, mitosis kicks in, embryo, mitosis kicks in, to being a baby, mitosis kicks in, to being an adult, mitosis kicks in. So this is all mitosis. This is meiosis. Okay, so let's talk a little bit about chromosome numbers. Chromosome number in mitosis, it stays constant. That means that all of your what we call somatic cells, non-sex cells, meaning cells that are not sperm or egg, they have a constant chromosome number. All of our cells in our body, with the exception of sperm and egg, have 46 chromosomes. As a human, 46 is our total chromosome number, or what we would call our diploid number. Your parent cell creates two genetically identical daughter cells. So if we're talking about like cells in our eyes, our heart, our hair, our leg, our kidney, any cell in our body that is not a sperm or egg, they have 46 chromosomes in them. They have the diploid number. They all have a genome from your biological mom and a genome from your biological dad. Haploid plus haploid make diploid. Homologous chromosomes of 23 pairs to make 46 chromosomes. So, let's talk about a different species. If a parent cell has 16 chromosomes, the two daughter cells it's going to make will have how many chromosomes each? Give us a look over for a second. Think. Maybe look back at your notes if you're like, wait, what? What is this question? In this question, 16, is that a diploid number or a haploid number? Good, it's a diploid number. So if we're talking about mitosis, this undergoes mitosis. We're going to talk about the different phases. There's a lot of little steps in here. The parent cell has 16 chromosomes. How many are each of the daughters going to have? 1, 8, 16, 32, or 64? Good, 16. So correct answer, 16. Remember that mitosis before mitosis happens, the DNA replicates. We get 16 times 2, which means you get a 16 going here and a 16 going there. I'm a visual person, so like when we have these questions, you can write on the exam, you can like map this out. All right, eukaryotic chromosomes. Eukaryotic chromosomes, the DNA takes different forms depending on what the cell is doing at any given time. When the cell is not dividing, it's just doing its everyday things. Like for example, if we're talking about islet cells of the pancreas and they're making insulin, they are in a chromatin fiber stage of DNA. The DNA is unwound in certain areas, especially the gene for the production of insulin is being read, and it's going through a process of making insulin. So we have that loose and free inside of the nucleus. We cannot, with a cell a cell not dividing, you cannot see the DNA with your eyes. It looks like a solid ball. Are we going to talk about what? No, we're not going to get that far. Yeah. When a cell is undergoing cell division, this loose strands of DNA don't need to be read. They're not going to be making important things for the cell, so they wind up really, really tight. And what we then see are these linear pieces. Remember the karyotype? You saw 2 number 1s and 2 number 2s and 2 number 3s. We call those chromosomes. We can physically see them with our eyes underneath the microscope. You will today see them. When you have chromosomes, the way that DNA that's all like big, loose pieces, it winds itself around these special kinds of proteins called histones. The histones give them some organization. We're just talking about very basic level of organization here. We're not going to get into all of the details of this, but the histones form a structure called a nucleosome, and each of the nucleosome parts, each of the kind of like swirls and making our chromosomes, they are linked together by what we call linker DNA. I like when biological terms make sense. So you have pictures of this below. That what happens with our DNA is that our DNA will be kind of loose when it's not dividing. You might need to read sections of the genes, like I said, like in a pancreas cell. If you're making, at that time, you're making insulin, this area might need to be read, so that opens up, and then you can replicate that. But when you are dividing, what you want to do is you want your DNA to be really, really tight and compact. And so what you have, you can see these little yellow balls. There's a lot more detail. If you ever take an advanced cell biology class, you'll learn about real good details about all of this and this process. But essentially, you have these little like kind of balls that the DNA wraps itself around. And you have each of these layers is called a nucleosome. 8 histones make up a nucleosome. And the linking part between each layer is called a linker DNA. You're going to look at this underneath the microscope. So let's just think about what I talked about. That what the difference between a chromosome description and chromatin description was. When we take a look at all of these cells, if I'm looking specifically at this, would you hypothesize, based on my descriptions, that this cell here is showing its DNA in the form of chromosomes or chromatin? Good, chromosomes. Chromosomes when you can see individual strands. So here you see the individual strands. Everybody in these 2 rows right here, all of these cells are showing their DNA in chromatin. The DNA, you can't really see individual pieces in all of these cells here. So we call this chromatin, chromosomes, chromosomes, chromosomes. In addition, the ends of our DNA have little hats or caps that are called telomeres. They give the ends of our DNA stability. As we age, these telomeres get shorter and shorter. So the protective caps of our DNA get shorter and shorter or smaller and smaller. And it's one of the many things that causes us to not perform as well as we get older, is the degradation of telomeres. Every time our cells replicate, you lose little bits of your telomeres or those protective caps on your DNA. And as you get older, things change and they don't work as efficiently. There's a lot of other reasons for that, but this is one of the many reasons that causes aging, is the shortening of your telomeres on your DNA with every replication. One of the things that chromosomes are bound to each other in the middle. When you have your pieces from biological mom, biological dad, when they the different pieces of DNA come together, excuse me, not the mom and dad. When you have individual pieces of DNA in your cells, you have them bound together and connected by what we call centromeres. whenever one of your pieces of DNA, a chromosome replicates. So if you have in your body, one of your cells in your body, the DNA is going to replicate. Your number one chromosome, let's say one of these one chromosomes, it makes an identical copy of itself. It temporarily gets stuck together and it looks almost like a little X, like this. And so you have your original DNA piece, your replicated chromosome. They get stuck together in the middle by cohesion proteins. This area is called the centromere. And then eventually they are going to split apart. So it's just a temporary attachment site. It's made of little microtubules, little like tubey pieces that hold it together, and then they slide to allow them to come apart. We only see all of these during cell replication. So here, this is the centromere area. It is made up of cohesion proteins. When this chromosome replicates, it gets what we call a sister chromatid. The centromere area made of cohesion proteins. At the end of cell replication or division, splits apart. One of these goes into one of your daughter cells, one of these goes into the other daughter cell. A lot of times students are like, what in the heck is the difference between the term chromosome and chromatid? This first one is called a chromosome. When DNA linear pieces are alone, they're called chromosomes. When your DNA pieces are replicated and attached, they're called sister chromatids. Chromatids are replicated chromosomes. Chromatids are replicated chromosomes. Okay, so a chromatid or sister chromatid is a replicated chromosome. Chromosome is one DNA piece, not replicated. Chromosomes are one piece, chromatids replicated and attached. I should say replicated chromosomes that are attached. Okay, chromatid, replicated chromosomes that are attached. Chromosomes, one piece. Remember this, because I know as you get into, like, you start studying and then you get all like, oh my God, what are these terms? Chromatid, replicated, chromosome, individual. Okay, back to chromosome numbers. Each species has a characteristic chromosome number or diploid number. Diploid is 2. 2N. N means a set of chromosomes. N, one set of chromosomes. N, one set of chromosomes, also means haploid. Haploid, N, one set of chromosomes, 2N, diploid, 2N, two sets of chromosomes, one from bio mom, one from bio dad. Every species has a characteristic diploid number. For humans, our diploid number is 46. We have 23 sets of chromosomes. 23 from bio mom, 23 from bio dad. 23 plus 23 is 46. Crayfish, crawfish, however you want to say it. Check this out. Here's an invertebrate species, an animal that doesn't have a backbone, has a very, very simple brain area. They have 200 chromosomes. So just because you have a lot of DNA doesn't mean that you're the best thing out there. Or maybe it does. Maybe they're like running the planet. I don't know. Diploid for humans is 46. Crayfish, 200. So when we're taking a look at humans, we have 46 chromosomes. There's 23 pairs. 2N equals 2 times 23. Formed in mitosis. Okay, oh, good. I do have that. Next. '23 is our haploid number. N, 23 plus N, 23, 2N. 23 plus 23 is 46. Makes our 2N times 23, 2N. Of our 46 chromosomes, 44 of those are what we call autosomes, non-sex chromosomes. And then we have 2 sex chromosomes. If it all goes well, you have 2 X's for biological females, and for biological males, you have XY. So everybody has 46 autosomes and 2 sex chromosomes. This karyotype here, what kind of species does it show? Yeah. They have 48. They're close. Yes, they share about 95.5% of the same DNA as us, but they do have. So this one would be the chimpanzee. We have 46 chromosomes, 44 autosomes, and 2 sex chromosomes. So yes, good. So in this case, it is a human, and good that you guys can see all the hands that were up. When we look at the last pair of chromosomes, we have an X and a Y. So this karyotype would be a biological male. We saw before a biological female. One of the things I want to point out is that, like all of these autosomes, so pairs 22 to 1, all of these are called autosomes or non-sex chromosomes. The X and Y are called your sex chromosomes. Are homologous pairs. Each of these is a homologous pair. They are about the same size, same shape, have the same gene banding pattern, except for X and Y. The Y chromosome over time has been shrinking. It does not carry nearly as many chromosomes. All of the genes on the Y chromosome, they function to make a biological male different than a biological female. They don't carry any traits that equate to something outside of making sperm or parts of the male reproductive system, whereas the X chromosome codes for a lot of traits that have nothing to do with our sexuality, but the Y does not. So that's why the X chromosome is bigger. It's also why males have to have 1 X chromosome. The way that our blood clots, very key genes are found on the X chromosome. Okay, so haploid number again. Haploid is 1 set of chromosomes, or N. The only cells in a body that have the haploid number are sperm and egg. All the other cells in your body are diploid. Haploid cells are formed in meiosis. Mitosis is for everything else. We said that the diploid number in humans was 46. So what's the haploid number? Good. 23 is the haploid number then. Humans, haploid number, N is 23. Remember our crayfish, we said their diploid number was 200. So half of that, 100N plus 100N, gives a diploid number. The haploid number is 100. N is 100. In humans, if we take a look at a sperm or an egg, they have each 22 autosomes, non-sex chromosomes, and one sex chromosome. Remember the term sister chromatid, as we said before. Sister chromatid is a duplicated chromosome. It's duplicated and attached to its genetically identical chromosome. Independent strands of DNA are called chromosomes. So chromatid is a duplicated and attached chromosome. Chromosome is independent. It's its own little strand. Your picture below shows you sister chromatids. So your pictures below are chromatids, not chromosomes.
In the sliding canvas for G2, it said the organelles duplicate inside the skeleton and dismantled, and this one says for G1 that the organelles duplicate.
It could be different. Textbooks say different things. So we're not right now, like when you take a cell biology class, you'll look into that a little more. But for this, we're not going to focus on that. But thanks for pointing that out. Okay, so again, sister chromatids, replicated DNA. Sister chromatids replicated and attached chromosomes are called sister chromatids. So let's go through this again. Chromosome. This one's a chromosome. These are chromatids. These are each a chromosome. So chromosome, chromatids, chromosomes. All right, we're going to take a look at the cell cycle, mostly focusing on mitosis. Interphase of the cell cycle. It includes the G1, the S, and the G2 phases of the cell cycle. The mitotic phase includes cell division and cytokinesis. Cyto, that means it's the last part of the cell process to divide up the cytoplasm. And this again will be reflected further in your notes too. G1 is a growth phase, grows in size. Some of the organelles replicate here, some replicate on. The DNA duplicates in what we call S, or synthesis. Growth 2 kind of ties up all those loose ends. Mitosis, cell divides all the organelles, divides the DNA. Make sure that each of the new cells has 2 number 1s, 2 number 2s, 2 number 3s. It's very organized and orchestrated. Cytokinesis wraps it all up by dividing up the cytoplasm. So here you see the cell cycle. You can see that the cell spends a lot of its time in interphase. Interphase, a lot of it is spent, the cell does what it does. So if it's a heart cell, it might be contracting, relaxing, contracting, relaxing. At certain phases in a cell's life, it replaces itself. The mitotic phase is quick. It doesn't happen very long because you want the cells to get back to doing their jobs. And then on occasion, they replicate. If there are 12 chromosomes in an animal cell in the G1 phase of the cell cycle, what's the diploid number of chromosomes for the organism? So take a look at this. We said that it's 12 in G1. Now read on. What happens next? So think about this for a second. So in the G1 stage of the cell cycle, it's 12. What's the diploid number for the organism? Yeah, good, 12. Okay, so if it's 12 here, what'll happen here in synthesis, the DNA duplicates, 12, 24, 24, goes back to dividing that 24 into 12 for each cell. So it'll be 12, 24, 24, dividing up that 24 to get us back to 12. Interphase looks like this. When we're looking at interphase, the cell doing its everyday thing, it has a nuclear envelope. Its DNA is all kind of unwound and loose. It's got different organelles doing their jobs here. Under the microscope, when you take a look today under the microscope, you're going to see it kind of looks like, you can see it's kind of spotty because it's like a bunch of strands of DNA all piled up inside of that nucleus. So it looks like maybe like a mottled or tie-dye color inside the nucleus, more solid than you see individual strands. Also, here's another kind of cell, different kind of cell. The nucleus in interphase looks more solid in color. What is the event that always happens before cell division? The organelles divide in half. Did the organelles ever divide in half? Do you want half organelles or do you want copies of organelles? Yeah, you don't want them in half, so that's ridiculous. So think about what it's saying. The cytoplasm undergoes cytokinesis. Does that happen before cell division or at the end? At the end, good. RNA winds into DNA. Have we even talked about RNA yet? So that should be a clue like, nah. The DNA produces proteins. This we're going to talk about later. That happens during normal cell process. The DNA replicates. Yeah, okay. So DNA replicates. Okay, let's take a break. Why don't you take like 8 minutes, come back at 1:30, and then we'll continue on. So everybody take a break. In the least, get up and stretch your legs.